Hello guys, welcome to the knowledge domain, the concept of life sciences and biotechnology. In today's lecture, we are going to learn the DNA, how it forms the chromosome, and how it organizes itself to fit in a small nucleus, or in a simple word or a simple expression, the chromosome folding. So this is the one strand of the DNA, deoxyribonucleic acid, that is the genetic material of a lot of organisms, though we find the RNA as a genetic material in some of the organisms like viruses. So here, this is a nucleotide that is composed of a phosphate group, a deoxyribose sugar, that is pentose sugar, and a nitrogenous base. Here it is thymine. So this is the 5' terminus. The 3' OH groups of this pentose sugar, it forms a diester bond with the phosphate group of the subsequent ribose sugar of the nucleotide. Similarly, the 3' OH of this nucleotide, it forms a phosphodiester bond with the phosphate group of the subsequent nucleotide. Like this, the DNA runs from the 5' phosphate end to the 3' OH. So this is one strand of the DNA molecule. So these nitrogenous bases, they form the hydrogen bonds, like adenine forms a double hydrogen bond with thymine, and guanine forms a triple hydrogen bond with the cytosine. So the complementary DNA strands run in the opposite directions, and it forms a double stranded DNA helix. Like this is the 5' to the 3' strand is running, and the complementary strand is running in the opposite directions. And there are the triple bonds and the double hydrogen bonds between the nitrogenous bases of the nucleotides. So each of the color is representing the nucleotides is depicted. And this is the helix. This is just a flat structure or the two-dimensional structure. In this image, we can see that the double-stranded DNA organizes itself into a helix structure, and one turn of this helix, there are the 10 nucleotides, and the distance between the ends of this one turn is 33.2 angstrom, and the distance between the two nucleotides is 3.32 angstrom. This DNA helix has a major groove and a minor groove. The diameter of this DNA helix is almost 20 angstrom in length or 2 nanometer in length. So whatever DNA double helical structures we usually study as a template are the B form of the DNA that is usually found in the sodium salt, or this is a sodium salt of the DNA because DNA are the negatively charged, so these are stabilized by the sodium salts. And the sodium salt concentration is very high, so the humidity is around 92%, and the DNA organizes itself into a B form of the DNA. So in this B form, the length we already discussed, 33.2 angstrom. There are almost 10 nucleotides per turns of the helix, and the inclination of base pair from the horizontal degree is minus 1.2 degree. So when the sodium salt concentration decreases, the humidity also decreases, and the DNA organizes itself into A form, where the one helix turns is almost 24.6 angstrom in length. There are 10.7 residues per turn, and the inclination of base pairs from the horizontal degree is 19 plus 19 degree. There is a Z form of the DNA where the one helix turns is almost 45.6 angstrom in length. There are 12 residues per turn, and the inclination of base pairs from the horizontal degree is minus 90. So that is a double-stranded DNA helix. That double-stranded DNA helix, that is 2 nanometer in diameter or 20 angstrom in diameter wrap itself into the 8 molecules, histone molecules. There are 4 types. Each of them has 2 molecules each. H2A, H2B, H3 and H4. These are called the core histone molecules. And the DNA nucleotide that is attached to this core histone molecules is almost 146 nucleotides. Okay. And these 2 nucleosomes has a linker DNA and it is associated with another histone molecules called the H1. There is a post-translational modification of these histones, or we call it the epigenetic modification of the histone molecules, like the acetylation of the lysine, methylation of the arginine and the lysine, then phosphorylation of the serine, and ubiquitinylation of the lysine residues. So this is one nucleosome. So this nucleosome further forms a helix, and this form of the folded DNA is called a solenoid structure, or the 30 nanometer diameter organization of the chromosome. That we'll discuss in slightly more detail in the following slides. Okay. So as we discussed earlier, the core histones are the H3, H4, H2, H2B, and the linker histone we discussed is H1. And there are 2 other types for the linker histone, H1 point or the H5. And the post-translational modification or the epigenetic modification we discussed are the acetylation, methylation, phosphorylation, the ubiquitinylation, as well as there is a summoylation of the lysine, ADP ribosylation of the glutamate, deamination of the arginine to citrulline, proline isomerization from the cis to the transforms. So this is the 30 nanometer solenoid structure we have discussed in the previous slides. This solenoid structure further form a helix to form a 300 nanometer fiber structure. That we will discuss in the following slides. So before going there, this is the size of the haploid genome. So one genome where in the gametes we have the haploid set of the chromosome. This is important in calculation. So we'll focus on the Homo sapiens. So we have 3.2 billion nucleotide bases in a haploid set of chromosome. It will constitute, if we put each of the chromosome, whatever 23 means pairs we have, means only 23. In one gametes we have 23 chromosomes. So if we put these 23 chromosomes end to end, the length will be almost 1.1 meter. So this length of the chromosome, it packed itself into the nucleus. Okay, that we are discussing. So we know the double-stranded DNA helix, 20 angstrom or the 2 nanometer in diameter. Then this is a nucleosome. So the nucleotide attached to the core histone molecules is 146, but in each nucleosome, the average number of the nucleotides is 200 in length, and the diameter of this nucleosome form of the chromosome folding at 11 nanometer. This is also called string with beads nucleosome. As we can see here, there are the beads-like structures. Each nucleosome looks like a beads. Then this string with the beads, it further organizes itself so the form a helix that is 30 nanometer in diameter, that also we have discussed, and it forms the organize itself as a loop radiating from a central scaffold of the protein, and a diameter of this form of the structure is 300 nanometer, and it further organizes itself into a 0.8 micron as a mitotic chromosome. So this 30 nanometer fiber, there are the 6 nucleosomes arranged around an axis. Okay, and it looks like this is one ton where there are 6 nucleosomes, and it looks like a solenoid. So what is the base we are discussing? Base pair, DNA double helix, we know there are 10 nucleotides. Nucleosome, there are 200 base pairs for each nucleosome or the each unit, which is the fundamental unit of the chromosome folding. So there are 100 per turn, so there are 2 turns, so 200. That also we have discussed. In the 30 nanometer, we have the 6 nucleosomes. So 200 into 6 is equal to 1200 base pairs. Then in the loop that it arrange itself around a central scaffold protein, there are 50 helical turns per loop. So 50 into 1200 equal to 60,000 nucleotide base pairs. And in one chromatid, there are such type of 2000 loops. So 60,000 into 2000 equal to 120 billion base pairs. So one chromatid at 120 billion base pairs. Okay. So in the one of the previous slide, we have discussed the human genome has 3.2 billion base pairs, which is 3,200 million base pairs. So 3,200 million divided by the 120 per chromatid is almost 27. It means 23. Okay. So each chromatid, it contains almost 120 million base pairs. So we have the 23 sets of the chromosome. So this total number of the nucleotide is 3.2 billion nucleotide base pairs. So what are the components of the eukaryotic genome? So we know the protein is synthesized from the sequence of the DNA that is called the exons, and this is the coding sequence of the DNA, and the place from where the protein information is not present is the introns. Okay. Then there are the DNA that encodes the RNAs, but those RNA do not contain the sequence information of a protein. These are called the non-translated RNAs. So those are the RNAs, but they do not contain the sequence information of the protein, like the small nuclear RNA, small nucleolar RNA, then the 7 signal recognition particle RNA, telomerase RNA, then X inactive specific transcript RNA, small regulatory RNAs. Along with it, there are the non-repetitive intragenic non-coding DNA. So the coding can be a protein encoding portion of the DNA, or it is a coding region of the DNA that codes RNA, but these RNA do not contain information of the sequence of the protein. Like ribosomal RNA, it helps in the protein synthesis. Transfer RNA also, it helps in adding the specific protein, but it do not contain the sequence information. They only help in recruiting specific amino acids to prepare a polypeptide chain based on the information available in the mRNAs. Okay. Then interspersed repetitive DNA, pseudogenes, are the stage of the DNA that contain the information of the protein, but due to some reason they are defective and they cannot express themselves. Okay, so those are the pseudogenes. Then there are short interspersed element, also called the sign. There are the ALU elements, which contain the internal promoter so that they can initiate transcription, reverse transcription, or the reintegration. And there are almost 1 million copies. Then there are the mammalian wide interspersed repeats, also called the MIR families. Average length is 130 base pairs, and there are 400,000 copies. Then there are the long interspersed elements, in short form called line. So the line 1 family, average 800 base pairs. This is almost 200,000 to 500,000 copies. Line 2 family, average size is 250 base pairs. It is 270,000 copies. Then the retrovirus like elements, the average length is 500 to 1300 base pairs. there are 250,000 copies, and DNA transposons, this is variable in size, average is 200 base pairs, and there are 200,000 copies. Okay, the others tandem repetitive DNA like ribosomal RNA are it is present in the 5 clusters, and in each cluster there is a 50 tandem repeats, and these 5 clusters, each of them is located in one chromosome, so it is found in the 5 different chromosomes. Then there is a transfer RNA gene, which actually recruits the specific amino acids according to the information available in the mRNA. It is found in the multiple copies along with the several pseudogenes that we already discussed what a pseudogene is. Then the telomere sequence. This is several kb. The total length is several kb, and each repeat unit is almost 6 base pairs in length. Then there are mini satellites, also called the VNTR. VNTR means variable number tandem repeats. So the total length is 0.1 to 20 kb, and each repeat unit is almost 5 to 50 base pairs, and they are mostly located close to the telomere. Then there is a centromere sequence, which is also called the alpha satellite DNA. So it is the 171 BP repeats. It binds to the centromere protein. Then there is the satellite DNA. It blocks of 100 kbp or longer of the tandem repeats, and each repeat unit is 20 to 200 base pairs, mostly located close to the centromeres. And mega satellite DNA, this is the block of 100 kb, means total block, and each repeat unit 1 to 5 kbps in various locations. Okay, so in this one you can see AGAC. So it is the mirror AGAC. So C is C, A is slightly distant, G is slightly distant, A is the farthest one. Like it is the GG is close, TT. So this is they are mirror to each other. This is called the mirror-like palindromes. Similarly, in this one, there is the complementary DNA. You can see GGAT, and in the complementary strand, in the opposite direction, GGAT. Like this, it is ATCC. Here also you can see ATCC. This is complementary to each other. And this is repeat. This is called the inverted repeats, because the repeat you will find in the opposite strand. So this inverted repeats, it forms a hairpin. You can see it here, GGAT. Okay. So here also in the complementary strand, here this is the ATCC. So GGAT, ATCC, you will see this is a hairpin. So when there is means almost 5 to 6 nucleotide present in the center of the inverted repeat, it will form a stem and a loop-like structure. This is the other arrangement of the chromosome. So next is how the bacterial DNA supercoiling happens, because in the one of the previous lecture we have discussed the prokaryotic nucleus. It is not called nucleus, it is called the nucleus because they don't have a nuclear membrane. So how the genetic material of the prokaryotes organize themselves? Usually this is called the nucleus because they don't have a nuclear membrane. So they are circular in nature. This is the left-handed coiling, which is almost each coil is 200 nucleotide in length, and this is called the negative supercoiling. To get a better view of it, so this is the left-handed coiling, it results in the negative supercoiling, and this is the right-handed coiling that results in the positive supercoiling. Okay. So like in the eukaryotes also, we have discussed that the loop of the DNA, the chromosome is possible because of there is a scaffold protein, or it forms the loop around the scaffold protein. Similarly, the prokaryotic DNA also organize itself around the scaffold. So there are 50 giant loops of the supercoiled DNA around the protein scaffold.